Ja, man. Dus ik kan de instelling gekomen ook zeker?
Wat zijn
Is er dan de instelling gekomen ook niet? Hoe ziet dat er ook uit?
Ja, dat is goed. Dat mag zijn. Stop dat je dat bakje er niet af van trekt.
Zeker niet blijven hangen.
Ik ben bang dat in internetkabel dat er gaat van trekken.
Oeh, hij zit er gaat toch meer lengte?
Ja, maar dat zit helemaal in de knoop. Kun je zien of dat hij ontvangt?
Nee.
Nee?
Nee.
Jan had gevraagd om hier de chauffage aan te zetten. Het is een bank op haar. Ja.
Ben je nog wel aan het livestreamen?
Ja.
Ja.
Nee, het zit wel traag.
Anders toch die hand is aan de ik ben schijf in ieder geval.
Oh, dat is al belangrijk.
Een halve infinity. Iets zo? 15 seconden?
Is het zo lang?
Ja. Ja, dat is het. Nou, toch een kleine 20, hè?
20 seconden. Dat dan nog. We hebben nog aardig geweest.
Ja, wat al bloot.
Nog maar ons.
Nee. Als ik je dat doe, je weet dat dat ging gebeuren? Ik zeg met die niet meer gebeurd. Dat is lekker langer, hè? Want
Wat is er met die lieve gedoe?
niet. Misschien maar over.
Wat is dat?
Er zijn al 6 mensen mee aan het volgen.
Succes. Allee, ga maar wachten. Kom, dat wil iemand anders zeggen. Maar je zit er natuurlijk ook tussen, voor die 6 mensen.
Ja. Allee.
Bedankt. Ja, doe maar. So, welcome for this last contact moment of comparative genomic analysis. So this is the third and the last contact moment. We will discuss 3 parts today. The first part is multiple sequence alignment, which is an introduction to the second part, being phylogenetic tree construction, and then we have a last part that is about genetic variation and human disease. Let's start with multiple sequence alignment. First, we can ask ourselves, what is a multiple sequence alignment? And we can see that a multiple sequence alignment is an alignment of 3 or more sequences. You should see this as an extension of pairwise sequence alignment, in which we try to create columns of homologous residues. Being homologous, they are derived from a common ancestor. So that we know that on every position in that multiple sequence alignment, we can actually see how evolution has been taking place. Why can we perform these multiple sequence alignment? This is because we know that our biological sequences are part of a family. This can of course be a gene family within an organism, so related genes. These have a name. How can we call these related genes in an organism? Paralogs. Yes, so paralogs. This can be sequences within a population. We call these polymorphic variants, or we can call these alleles, so the different alleles that are possible on a location. Or this can be genes in other species, and these we call orthologs, or a combination of these 3. So it's possible to bring them all together into one multiple sequence alignment, and to use this multiple sequence alignment to actually create a phylogenetic tree. If we create a multiple sequence alignment, for example, a protein multiple sequence alignment, we have amino acid residues aligned. How can we align these residues? This is based, of course, on properties of the amino acid residues. And this is different than, for example, nucleotide sequences, where we cannot use the properties of our nucleotides to align these sequences. So there we only take into account identity, but here we can also take into account similarity by using scoring matrices to help align our sequences. There are some anchor positions, for example, some conserved residues like cysteines, which form disulfide bridges, but also, for example, tryptophan residues can be very typical, aligned in our multiple sequence alignment, and show that this is actually a conserved residue in our alignment. There are, of course, other properties that we can also take into account to create these multiple sequence alignments. If we look at the different algorithms that are available to create this multiple sequence alignment, we have a first alignment method, which is an exact method, which is actually some kind of extension of what we saw with Smith-Waterman or with Needleman-Wunsch alignment. Of course, that is for pairwise sequence alignment, but this follows the same principles, meaning that at the end of our exact method, we will have the multiple sequence alignment with the highest possible score. So the optimal multiple sequence alignment. After these exact methods, which we can only use for a limited number of sequences, we have progressive alignment methods, and actually all the methods that are not exact are progressive methods. If they take into account some possible suboptimal solutions and try some iterations to increase the performance of our alignment, we talk about iterative methods. And then there are also consistency-based methods, which, for example, make use of profiles, which we saw in the previous lecture. So this can be POSSUM, so position-specific scoring matrices, or the Markov models, which we can use in these consistency-based methods. A last method that we can use is structure-based methods, and the structure-based methods are actually the best way, because here we use structure information, which is more conserved than sequence information, to aid our alignment. But all of these are, of course, progressive methods. If we want to choose an alignment method, we need to take into account the speed and the accuracy of our alignment method. Meaning that if you choose a progressive method, this will be a very fast method, but will also be a less accurate method. If you take, for example, an iterative method, we can actually try to combine both properties. So we can actually have a fast algorithm which also has a good accuracy. Examples of these, for example, for speed or muscle and MUFT, if you look at accuracy, we're looking at the consistency-based methods. So PropCons and tCoffee are the consistency-based methods, but they are actually slower than the other alignment methods. How does a progressive alignment method work? This is a very simple way to perform a multiple sequence alignment, and this is based on placing two sequences together in an iterative or in a progressive way. Meaning that we start with the two most related sequences, being the sequences with the highest pairwise sequence alignment score, and then we take a third sequence, which is next in row, and then a fourth and fifth, and so on. So in a progressive way, we will add sequences to our multiple sequence alignment. We start with the two most closely related sequences. We can actually try this. Therefore, we need to use Mega. I don't know if you have already Mega installed. No? Then I'll show you how to install Mega. So you can go to Google, and on Google you can search for Mega software. Mega stands for Molecular Evolutionary Genetics Analysis. You can choose the Windows or the Mac version that is available, but we'll use the Mega 7. So not the Mega X, but the Mega 7 version of that software. You can download and install it, and I'll just show you for this exercise how you can use it. So in the meantime, you can actually try to install the software. If you start Mega, you get this window with different possibilities on top. As you can see, all the possibilities here to perform a phylogenetic analysis are available. First, we need to start with the import of our sequences. Of course, therefore we need our sequences, and here we have 5 divergent globin sequences that we would like to analyze. We have 5 different accession numbers, which we can use to look for sequences in the NCBI protein database. If you want to retrieve all sequences at once, you can copy all the accession numbers. You can go to NCBI protein. And at NCBI protein, you can paste all the accession numbers separated with a comma, which means that this last AND should be changed to a comma. If you next submit your search, you will get 5 hits, being 1 hit for every accession number that we entered to search with. Now you can select the FASTA sequence for every entry individually, or there is another possibility, you can select to format this in FASTA text. And this way, this will actually create a multi-FASTA file of these 5 sequences, so that we can actually download these 5 sequences at once. If you select FASTA text, this will be our multi-FASTA file. This multi-FASTA file we can save in a text editor, so a plain text editor. So Ctrl-C, Ctrl-V. And this multi-FASTA file we can give a name. I'll save this as. Then I'll go to comparative genomic analysis, data, and I'll save this as divergent globins.fasta. Remember that if we save this as divergent globins.fasta, that we need to remove the .text extension. So do not use the .text extension, and then you can save it. If you would like to use this file in Mega, we can import this with the alignment button. Is this big enough or should I set it a little bit bigger? Big enough? Or maybe for people from home, I'll try to set it a little bit bigger.
Should we install it in Linux as well?
You don't need it in Linux. No, you don't need it in Linux. Yeah, it's a little bit bigger. Now you can import your file, therefore you select the Align button, and you select to edit or to build an alignment. So Align, Edit, Build Alignment. The option that you can use is to retrieve our sequences from a file. Because we just saved them into a FASTA file, we can now retrieve the sequences from a file. Here you can browse for these sequences. So here I can find my divergent globins.fasta. I can select this and I can open this in this is called the Alignment Explorer. In the Alignment Explorer you can change the names of your sequences if you would like that, so you can make them shorter or remove some things or rename them. You can also, for example, assign a group. If you assign a group, this can be visualized when you, for example, create a phylogenetic tree. Members from the same group will be indicated in your tree. As you can see now, these sequences are just aligned based on position numbers. So if you are the first amino acid residue, you come on position 1 of my alignment and so on, meaning that at the end of my alignment I will have gaps because these sequences are not aligned. Of course, this is not a multiple sequence alignment, because here we just place it at the beginning of my positions. If you want to align our sequences, you have two possibilities in Mega. You have, for example, here, so for this progressive method, we can use Clustal W. So Clustal W is an example of a progressive alignment method. Here you can select the W from Clustal W. If you select the W here, and select to align proteins, it will ask you to select all sequences, okay, and it will perform an alignment. You can see that, for example, here the blossom matrix is used to create this multiple sequence alignment. You select OK, and you will get a multiple sequence alignment. As you can see, the gaps at the end are a little bit less, and if you scroll, you will also have some internal gaps. These are 5 divergent sequences, which mean that they are diverged a long time ago in history. So that's why they have these gaps and they have actually not that good alignment. If you look, for example, at the positions that are retained during evolution, we have these conserved positions which are indicated with a asterisk. So here we can actually find which positions are conserved in our alignment. For example, here we have a histidine which seems to be conserved, but the number of identical residues in the multiple sequence alignment is very limited. So this is for Clustal W. If you would like to use this multiple sequence alignment, for example, to create a phylogenetic tree in the next step, you need to send this data to the other applications. We can send this data to the other applications by using the data phylogenetic analysis button. So if you go to data and you select phylogenetic analysis, it will send these results, this multiple sequence alignment, to the rest of the applications that are available so that you can use them for analysis. If you select that button, you will see that this changes a little bit, and now you can use the different tabs that are available here to analyze your sequences. Of course, if you would like to create a phylogenetic tree, it is important that you first create a multiple sequence alignment. So first you will use Clustal W or muscle to align the sequences, then send it for phylogenetic analysis, and then you can use the different tree construction algorithms that are available. So this is important. So this was the first example for these 5 divergent sequences using Clustal W. Next we will see another method that we can use, but there are some remarks that we should make. One problem that we have with these progressive methods is that we take two sequences together, but if we have a mistake in the first two sequences that we bring together, we will feel this mistake by adding other sequences in a progressive way. Which means, once we insert a gap, being correct or incorrect, this will always be a gap. We cannot remove it anymore. So there's no correction of mistakes. If you make a mistake, it will keep the mistake until the end. And this is different for example, iterative methods. Iterative methods have some possibilities to remove mistakes that are made during the progressive alignment of our sequences. Therefore, the algorithm has a different insertion penalty and a deletion penalty, because a wrong insertion or deletion is less good than one of the other. Which one is worse? A wrong insertion or a wrong deletion? So normally you have an alignment, it's a perfect alignment, but now the algorithm introduces an insertion or a deletion that is not correct. It should not be there, but it's still in the alignment. What is worse? A wrong insertion or a wrong deletion? Insertion, why? It's correct. A wrong insertion is worse than a wrong deletion. Because once you have a wrong insertion, you will see the insertion in your multiple sequence alignment. And if we align a next sequence in our progressive alignment method, every time that we meet that insertion, we need to provide a deletion to compensate for the wrong insertion. Which means if I have an alignment, for example, A, C, G, T, A, C, G, T, and this is my wrong insertion, the next time that I align my sequence, I will have A, C, G. This was wrongly inserted, so I'll have to provide a gap, so a deletion to compensate for this insertion. And then I'll go on with the rest of my sequence. If I align the next sequence, because it's a progressive method, I again will align. I'll see my wrong insertion, so I'll have to compensate with a deletion. So one insertion has to be compensated with multiple deletions. And therefore, an insertion is... Ik weet niet goed dat ik het zeg in het Engels, slechter than a deletion. Is more bad? Worse. It's that simple. It's worse than a deletion. Because we need to compensate for that over and over again. If we do it the other way around, for example, we have an alignment, and in our alignment I have a deletion, but the deletion is not correct. So for example, A, C, G, T, I have a deletion, or I provide a deletion, but it should not be there. The next time that I align my sequence with my progressive method, so I'll align my next sequence, I can just insert it without having to provide a penalty. So I have no penalty for correcting for a deletion, a wrong deletion, of course. And that's why the insertion penalty is actually bigger than the deletion penalty. I hope you understand this a little bit. This is a problem for progressive alignment methods. This is not a problem for example, iterative methods, because there they have the possibility to correct these mistakes. Iterative methods think that they calculate a suboptimal solution using a progressive alignment. So they try the alignment method, for example, that is used by cluster W, but they try to adjust the alignment in an iterative way, which means that they will have multiple cycles of trying to solve these problems, trying to create a better alignment, and if you have a better alignment with a higher score, it will replace the original alignment that was created using the progressive alignment methods. Most of the times this will have to do with the order in which we progressively align our sequences. And this way we will split up the multiple sequence alignment into different parts, try to realign the different parts individually, and then try to bring it back together. So this is a possibility that we can do with these iterative methods. In this way, our problem, once a gap or once an error, always an error, is solved. So these methods should be better than the progressive alignment methods. An example of an iterative method is muscle, which is also available in Mega. The other methods are not available in Mega, so we only have cluster W and muscle. That's why we choose one of these. But there's no consistency-based method or a structure-based method that we can use, but these are available on the web and in other software packages. Is Mega installed? Yes? Did you download the 5 divergent globin sequences? No? So use the accession number separated with a comma, go to NCBI protein, paste it there, and then show the output format as FASTA text. And there you can download it as one FASTA file. If you have downloaded it, you can import it in Mega, and then you can try to align the sequences using muscle. So in Mega, I can close this to start over again. So I'll close my data, and I'll show it again. So we go to Align, Edit Build Alignment, and retrieve sequences from a file. Here we can open the divergent globins.fasta, and in the next step, we can align these sequences using muscle. So here we have the muscle icon to align the sequences using muscle. We use the default settings of muscle, meaning that if you're not sure if these are the default settings, we can restore the defaults. And then we can select compute.
Did you, for the cluster, did you change the default?
No.
Because when I tried it, there was another matrix instead of blossom. I changed it to blossom, but it was the third one.
The gonad.
Yeah.
There's something wrong with my mouse. If you look at the alignment, we have the same problem as before. So we only have a couple of identical residues. We can find our histidines back here. But for the rest, we also have gaps that are introduced in our multiple sequence alignment. Of course, if we perform a phylogenetic analysis, the positions that have gaps will not be used for the analysis. So this will not be used. Only positions that have a residue in all sequences will be used, because you need to calculate distances between sequences. And there's something wrong with my selection. Did that work?
Nee. Het is een probleem met die vaste apparaat niet werkt. Daarvoor dat ik een mailtje had gestuurd.
Ja, en is dat niet gelukt om op te lossen?
Nee, en op Mac lukt het niet, maar op als je het dan via Windows doet, lukt het wel.
Op een keer je bestand. Je ziet, er zit dan nog altijd opmaken. Hè. Voorkeur en hier platte tekst. Dus een nieuw document met als platte tekst. En als je nu een nieuw,
Ja, ik zal het nu aanmaken.
als je er een nieuw document gaat pakken, gaat hij normaal zijn als platte tekst. Dus omdat hij er opmaak insteekt en door die opmaak herkent hij het niet als vaste document. En dan mag hij niet doen. Dat is inderdaad het probleem opmerken, omdat het er anders is. Bij klantblok is dat standaard platte-tekst. If we have a look at the fourth way, we have the consistency-based methods. Here we try to use information from multiple sequences to aid our multiple sequence alignment construction. Of course, this seems to be weird because we want to create a multiple sequence alignment, but we want to use information from multiple sequence alignment to aid that. Of course, here the information from multiple sequence or from multiple sequences is not yet available in our input sequences. So if you, for example, have 5 sequences that you want to align using a consistency-based method, the information from multiple sequences is not from these 5 sequences. This is information, for example, from a position-specific scoring matrix or from a hidden Markov model that is used to aid the alignment of our 5 sequences. So what actually happens is that we use one or all of these 5 input sequences and we search for profiles in a profile database. And there we use the information that we have in this profile database to aid the multiple sequence alignment method. This also makes use of deduction, meaning that if 2 sequences are related, for example, A is related to B, and B is related to C, that we can use this relationship to also align A to C. So we use the information from relationships that we know to create other relationships. This is called deduction. These seem to be very good algorithms, so for example T-Coffee or PropCons, but of course they are a lot slower. And the last one is a structure-based method. As indicated, if you look at conservation, and we look at, for example, nucleotide sequences, they are less conserved than amino acid sequences. So there is more homology in amino acid sequences. If you go to the next one, for example, we look at 3D structures, we see that these 3D structures are even evolving slower than the amino acid sequences themselves. So therefore, if you would like to align proteins, it's better to do that using the 3D structures than to use the sequences alone. Of course, there are some problems here, that not for all proteins that we have, there is 3D information available. So if we don't have 3D information available, we cannot use this information to align our sequences. There are some rules. For example, if we add homologous sequences to our input, this will actually increase the accuracy of our method. And this is useful because adding a homologous sequence can close the gap between 2 sequences. Meaning that if there is a certain distance between sequence A and B, so the distance between A and B is very big, it's very hard to align these 2 sequences in a multiple sequence alignment. If we add a homologous sequence, it is possible that this homologous sequence is more closely related to E and more closely related to B. In this way, this can aid the alignment of A to B with some intermediate step using sequence C. So it's always a good idea to add new sequences to our multiple sequence alignment, of course, if they are homologous. What else do we know? If the percent identity decreases, also the accuracy decreases. And this is, of course, useful, that there will be more mistakes made, there will be more possible alignments, and therefore we cannot always rely on the results that we have. For divergent sequences, for example, the 5 sequences that we have, it's actually better to use a local alignment method that only focuses on the parts of our proteins, of our sequences, that are more similar than these more diverged parts of our sequences. Examples of algorithms are D-align and LAGA. And this is different from what we use, and also from the default strategy, being that our multiple sequence alignment algorithms are most of the times global alignment algorithms. And you can also see this when we align our sequences using Mega. You can see that every residue is aligned, so this is global alignment. And this is different, for example, from BLAST, which is a local alignment algorithm. So here there's a big difference between the pairwise alignment algorithms that we use, which are most of the times local, and the multiple sequence alignment algorithms, which are most of the times global alignment algorithms. Another thing is the introduction of an orphan, and if we introduce an orphan, being a very distantly related member of the family, that we better use a global alignment algorithm. Because if we would use a local alignment algorithm, this would remove a lot of the alignment. So this would actually try to remove as much of the alignment as possible, and we would only have a very small alignment left. So therefore it's better to actually use a global alignment, because we will have a solid alignment of the more closely related sequences, and it will just try to place that orphan as a last step on that good multiple sequence alignment. And there are some obvious things if we combine multiple sequence alignments, so multiple sequence alignments, we better use an iterative method. If we have large extensions, it's better to use a local alignment than a global alignment, but in general we always have global, or most of the time we use global alignment algorithms. If we use multiple sequence alignment, we do this most of the time for short sequences, so for one gene, for coding sequence, for a protein sequence, and so on, but we do not do this for large genomic DNA sequences. There is a possibility to align these large genomic DNA sequences, especially in the case where we want to examine positive or negative selection. So if we want to investigate positive and negative selection between multiple, and here we talk about organisms, so we compare different types of organisms, then we can get an insight in what is positively selected and what is negatively selected. For example, here we have a multiple sequence alignment from a genomic sequence. This is my genomic sequence, and this is a human genomic sequence. And on that human genomic sequence, we actually try to align different types of organisms. So all these organisms are aligned in a multiple sequence alignment to my human sequence. You can see that some regions are conserved. For example, here, this seems to be an exon that is conserved in all organisms. Here we see, for example, an exon that is not conserved in all organisms. For example, if we look here, we can see that this exon in this range is not conserved in, for example, Xenopus tropicalis or the chicken. Here we have two different situations. One is positive selection, the other is negative selection that we can investigate. But which one is positive, which one is negative selection? So here we have conservation between, for example, chicken and humans. Here we don't have conservation between chicken and humans. Which one do we call positive selection, which one do we call negative selection during evolution? This one is called negative selection. Why?
It hasn't changed.
It hasn't changed. So this means that in evolution, between the speciation of chicken and humans, there have been mutations in this sequence. There have been mutations, but they are not retained. Therefore, there is negative selection for these mutations. On the other hand, here, this was once upon a time the same sequence. Then we had the speciation between chicken and humans, and today they look different, meaning that there are mutations on that position which are retained, which are selected for, and therefore this is my positive selection. So this is the difference between the positive and the negative selection that we can examine when we actually try to align these large genomic DNA sequences. This is an exercise. X-linked adrenoleukodystrophy is the most common inherited disease affecting peroxisomes. The disease is caused by mutations in the ABCD1 gene encoding alt protein, so alt-P. We will create a multiple sequence alignment of the alt-P family of proteins from human, so alt-P, alt-F, PMP70, PMP70R. These from mouse, which are called ABCD1, 2, 3, and 4, and from yeast, which are called PXA1, PXA2. in these proteins there seems to be a nucleotide binding site, and this nucleotide binding site seems to be conserved, and it's our goal to use these sequences to actually try to detect this nucleotide binding site in the multiple sequence alignment. Where can we search for these proteins? NCBI, protein, but there we will have a lot of information. So better ways to start at. You can start at taxonomy browser, yes?
Or gene.
Yeah. So we can start at the taxonomy browser and then go to NCBI gene. So I'll start at the taxonomy browser. The first organism that I'm interested in is human. Of course, this is most of the time not why we use the taxonomy browser. Then we can go to gene, and do we select subtree links or direct links? For humans, it's better to use direct, because we know that we have Denisova and Neanderthaliensis, which are in the subtree links. So here we can use direct links. And now we can search for these genes. For example, Alt-P. So I'll type and Alt-P. And you can see that this redirects us to ABCD1, which is the name of the gene. Alt-P is the name of the protein. So here I have ABCD1. I seem to have 1, 2, 3, 4. So maybe I have ABCD1, 2, 3, 4. And this is correct. So I have these 4 sequences. You can see that this is Alt-P. The other one that we have is Alt-R, which is this one, ABCD2, which is called Alt-R. And then we have PMP70, which we can find here. So this is ABCD3. And then we have PMP70R. It's called P70R, which is ABCD4. So we need the protein sequences of these 4. Yes, the protein sequences of these 4 genes. So I'll open them in a new window. And I'll go to NCBI reference sequences. So I'll start with ABCD1. I'll go to NCBI reference sequences. And there I can, for example, copy the NP number to CloudDoc. And this way I can see this, save the sequences in one action. I'll call it an action. I'll go to ABCD2, NCBI reference sequences, and I'll copy the NP number. ABCD3, NCBI reference sequences, and I'll copy. There are multiple isoforms. I'll take the longest isoform, so I'll take this NP number. That's the second one. If you don't know which isoform to take, you take the longest one. Always. And then ABCD4. And here we also have multiple isoforms, a lot of different isoforms available. A very large number of isoforms available. And here we can again select isoform A, so the longest isoform. Now I can do the same, but for another organism, for mouse, ABCD1, 2, 3, and 4. So I'll go back to my taxonomy browser, and in the taxonomy browser I'll search for musculus. Here, do I choose subtree links or direct links? Subtree, so here I want also the subdivisions of the different mice that we have available. And here I can search for and ABCD1. With a little bit of luck, we'll find ABCD1, ABCD2, 3, and 4, but I cannot find 4. So I'll open 1, 2, 3, and I'll search for 4. And 4. It's already open. Again here, I'll go to my reference sequences and I'll copy the NP number. So this is ABCD1. I'll do the same for ABCD2. I'll choose the longest isoform. ABCD3. Here there's no information available about which isoform is longer. I'll choose the first one, the isoform 1. Maybe I can have a look at the different domains. Yeah, I'll choose isoform 1. And then I'll do this also for ABCD4. There is only one isoform. And now I can do the same. This is the last one for yeast, which is called PXA1 and PXA2. So I'll go to my taxonomy browser. I can type in yeast. What is yeast called in Latin? Saccharomyces. Let's try that. Saccharomyces cerevisiae. Baker's yeast. Here we can also select the subtree links for NCBI gene. So Saccharomyces cerevisiae, Baker's yeast. And here I can search for PXA1 and PXA2. So I can find my PXA1. Can I find PXA2? Not directly. So I have to change this. And PXA2. Also here, I go to NCBI referring sequences and I can copy the accession numbers. Once you have all these accession numbers, you copy the list to NCBI protein. paste list here. And then you should normally get 10 results. I can format the output as FASTA text. And I can copy everything to, for example, Padlock or a plain text editor. And now it should work. Does it work?
Ik heb me ook aan want daar kun je dat er nog tekst achter.
Maar als je het opstaat, kun je maar aanvinken dat je dat nu mag doen. Ja. So again, here you can save this as, let's call this abcd dot FASTA, and I'll remove the dot text extension. Because these are the ABCD genes. I'll save this. Of course, we are interested in this presumed nucleotide binding site, so let's hope that we can find this presumed nucleotide binding site. So I go to Mega, and in Mega I will import my sequences. So I will retrieve my sequences from a file. If you look at these sequences, you will not find the presumed nucleotide binding site, because we did not align the sequences yet. And now we have two possibilities. We can align them using cluster W, or we can align them using muscle. Which one do we choose? This is normally an easy question. Muscle, and why?
Because cluster looks global.
This will also do global.
It doesn't correct mistakes.
It doesn't correct mistakes. So it has a once a gap, always a gap problem. And muscle actually tries to correct these mistakes, because it's an iterative method. So we use muscle to compute. And then I am interested in the presumed nucleotide binding site, which is GP something GCGKS. So GP something, my memory is not that good. So let's have a look if we can find it. I think it's here. So GPN GCGKS. GPN GCGKS. So it's here, and it's almost conserved in all organisms. So 4 of the 8 residues seem to be conserved in all organisms. So GPN GCGKS. Can you find it? It's on position 681. If this can help you. Do you find it? It's the same position? Perfect. So here we can actually identify, but you can see that there are some members. For example, here isoform 4, so member 4, and also here member 4 that seem to have some differences. That have most of the differences in this presumed nucleotide binding site. This would be a good example if we would like to use these sequences to create a phylogenetic tree to assign a group. For example, all human sequences should get the group name human, and so on. This would be a good way to test this. Of course, this is free to choose if you would like to do that. In the next step, we can have a look at molecular phylogeny and evolution. So we want to investigate, or we want to use these molecular sequence data to investigate the relationship between sequences, to invest the relationship between molecular sequences being on DNA, RNA, protein level, and so on. So we talk about evolution when we talk about the process of acquiring mutations and having selection to be able to select or not select for these mutations. So both have positive or negative selection. The phylogeny part is to actually determine the relationship between these, this can be organisms, between these components. Normally this is done on a morphological level. So most of the times this is, for example, in vertebrate organisms done with, for example, bones in the hand. So by comparing the organization of the hand bones, we can try to determine the phylogeny between organisms. Of course, we are not interested into this morphological characteristics, but we are interested into molecular phylogeny. So we will use the molecular data that is available to determine the relationship between sequences. If you look at our sequences, we can see that some sequences, for example, some proteins are very slow and evolve, or have a very slow evolution, meaning that they have mutations, but they do not change a lot over time. Other proteins are very fastly accumulating substitutions. For example, if I think about immunoglobulins, these are examples of proteins that are changing a lot over time. Another example of a very slowly evolving protein is, for example, histones. Histones are very important for the compactation, can I use that term, of chromatin in the nucleus, and therefore they are very conserved and they are evolving very slowly. There seems to be some kind of hypothesis, a molecular clock hypothesis we call this, which actually says that we can place the divergence time in relationship with the number of amino acid differences that we can see between protein sequences. This is the molecular clock hypothesis. In this way, we know for a gene family, or for a protein family, that if we have this number of differences, this corresponds with this time interval, in millions of years, for example. Of course, this is true for a gene family, for one family. But if we compare multiple families, we will see that the relationship between the divergence time and the number of amino acid differences is different for every family. Meaning that, for example, if we see 10 substitutions in protein family A, this can correspond with 100 million of years. If we see 10 substitutions in protein family B, this can already take into account to 1,000 millions of years. Just as an example. So there is a relationship between the number of substitutions and the divergence time, but this is different for every family that we have. This actually does mean that if we have a next time interval, that we will have a constant number of mutations there, so that there is a constant speed of evolution for our sequences. If you look at the relationship, this means if we plot the millions of years since divergence of our sequence against the observed number of amino acid changes, that this follows a linear trend for a protein or a gene family. So we see that there is actually a linear correlation, but this is different for different protein families. The difference that we see is, of course, based on the functional and the structural constraints that we have for these proteins. If we have, or if this molecular clock hypothesis is correct, we can actually use the time scale on our phylogenetic trees to not talk about distances, but to talk about divergence times. And this is why we want to investigate this molecular clock hypothesis, because normally if we create a phylogenetic tree, for example this one, and we look at the time scale that is indicated on that tree, we see that this time scale represents distances and not divergence times. If the molecular clock hypothesis is correct for the family members in this tree, we can actually use the time scale to indicate divergence times. And that's why we want to check this molecular clock hypothesis. Note that the speed of evolution that we have is dependent on the organism. We can have organisms that are evolving very fastly. Also depends on the family that we are investigating. For example, histones are very conserved, but then, for example, immunoglobulins are very fastly accumulating mutations. And of course, this is dependent on the function of the gene that we have. How can we test this clock-like behavior? We can do this with the Tajima's relative rate test. And the Tajima's relative rate test is available at Mega Software. What do we need for the Tajima's relative rate test? This is, of course, 3 sequences from the same family, but we need to have 2 closely related sequences, which are, in my example, sequence A and sequence B. And we need to compare this with an outgroup being a more divergent member of the family. We will have a look at the difference between A and C, and the difference between B and C, and in this way we will try to see if we have a constant speed of evolution. How can we do this? This is of course done with a statistical test called the chi-square test, and in this chi-square test we try to reject the null hypothesis of equal evolutionary rates. In this way, if we do not reject the null hypothesis, we can actually indicate that we have clock-like behavior. This is what we want to do. We'll do this with an example. In this example, we will use RBP4 from humans and from Felis catus. Do you know what Felis catus is? Cat? Catus? It's a cat. And then we will use an outgroup. And as an outgroup, we will use a predicted sequence. So it's not verified from an outgroup. Because if we want to investigate molecular clock, we will compare the speed of my human protein against the outgroup and the speed of my Felis catus against the outgroup. And in this way we can investigate if we have a molecular clock behavior, so a constant speed of evolution. And that's why we need this outgroup. If we do not have an outgroup, we cannot determine equal speed of evolution. So let's have a look at the sequence information that is available. We will start, for this analysis, we can start at the taxonomy browser. Taxonomy browser. Of course, for humans this is not necessary, but we can do this for Felis catus. So I'll search for Felis catus. And for Felis catus, you can see this is domestic cat. We can go to NCBI gene. At NCBI gene, I can search for RBP4. Here I go to NCBI reference sequences, and I can use the accession number to find the FASTA sequence. Here I will create the FASTA file, so the multi-FASTA file myself. So I'll copy the FASTA sequence of Felis catus to my text document. I'll do the same for RBP4 in humans, but therefore we do not actually need the taxonomy browser, so we can go directly to NCBI gene, and at NCBI gene we can search for RBP4. Normally, the first or one of the first hits should be RBP4 in humans. So there we do not need the taxonomy browser. So also here I go to the gene record. I select NCBI reference sequences. You can see that there are multiple isoforms available. And also here I will choose the longest one, but of course you cannot see it, so I'll use isoform A. So I can select this. I can select FASTA, and I can copy the FASTA sequence to my text document. This has to be on a new line. Next, I'll add my outgroup. The outgroup has an XP number, so I can use the XP number. I can go to NCBI protein. In NCBI protein, I can paste the XP number. It's removed, but we can have a look at the obsolete version. And we can see that this is from Crassostrea gigas, which is an oyster. I think it's somewhere Pacific oyster. Also here we can select FASTA, and we can copy the FASTA sequence to my text document. I can save this multi-FASTA file, and I can call this rbp4.fasta and remove the .text extension. Now I can go to Mega. I'll close my abcd.fasta, and I'll start with a new alignment, so AD build alignment. I'll again retrieve the sequences from a file, and now I can open my rbp4.fasta. If we want to determine molecular clock-like behavior, we can go to clocks, and here we can select Tajima's relative rate test. But the problem is that we don't have a file to analyze this data with, or we don't have data to analyze with this algorithm. Therefore, we need to send the alignment that we have here to Mega to analyze it with the Tajima's relative rate test. But first, before we can send it, we need to do something very important: align it. So before we can determine clock-like behavior, we need to align our sequences. We select muscle, align protein, compute. Now that we have aligned our sequences, we can select data, phylogenetic analysis. So we can send the results of our alignment to Mega for phylogenetic analysis. You can see that the icons have changed, and now we can go to clocks, Tajima's relative rate test. In Tajima's relative rate test, we can indicate 3 sequences. 2 closely related sequences, taxon A and B, and an outgroup. For this example, this is correct. So the order is correct, but you should check this. So the outgroup should be specified with outgroup, and then we have our 2 other sequences. You can see that this is amino acid sequences, and if we don't have data on position or complete data, we remove these positions. This is the last one. So we don't change any settings. And then we select compute.
Als dat niet klopt, kun je zelf
Ja,
weten.
je kan daar gewoon op klikken en weten. Ja. The good thing about Mega, and this is something that is very useful, is the caption below the results. So the captions in Mega are very good because they provide us a lot of useful information. Also, when you, for example, create a phylogenetic tree, the caption actually indicates all the information that we need to construct that tree, or all the information that is used to construct that tree. We can try to read this. So the equality of evolutionary rate between sequence A and B when comparison to sequence C, used as an outgroup in Tajima's relative rate test. The chi-square test statistic was 1.8 with a p-value of 0.17971. So this is my p-value with 1 degree of freedom. And then it actually explains what that means. If we have a p-value of less than 0.05, which is our threshold, this is used to reject the null hypothesis of equal rates between lineages. So if my p-value is lower than 0.05, I reject H0, and H0 says that we have equal rates between lineages. So that we have molecular clock behavior. In our example, we have a p-value of 0.18. This is not lower than 0.05. Therefore, we cannot reject the null hypothesis, and therefore we have molecular clock-like behavior. So we have equal evolution rates among lineages. In this example, we can therefore use the scale as a time measurement, and not as a distance, but as a time measurement. So therefore, these captions are very useful. Just read them and you know what the results are. So this is the Tashima's relative rate test. And the next step, we would like to create these trees, these phylogenetic trees. If you look at the tree, we have two different types of information. One is the branching order, being the topology, we call that, so the relationship between the sequence. Another one is the branch lengths. And the branch length, of course, indicate the amount of relatedness that we have between sequences. If we have molecular clock-like behavior, we can use the branch length as a time interval, so that we can know how much time we need to diverge these two sequences. The sequence that we use as input in our multi-FASTA file and in our multiple sequence alignment are at the end of our tree, and these are called OTUs. Of course, if we create a tree, we have a sequence A, we have a sequence B, but we also have internal nodes. And these internal nodes also represent taxonomic units, so a species somewhere in evolution. But this is extinct, so we do not have that species at the moment that we create this tree. Therefore, this is not an operational taxonomic unit. This is just an internal node which is inferred from the other sequences. Another thing that we have when we construct a tree is the possibility to create a rooted or an unrooted tree. There are algorithms that automatically create a rooted tree. There are algorithms that automatically create an unrooted tree. There are two possibilities for a root. I'll start first with explaining what a root is. So we can have, for example, this, which is an unrooted tree, and this just indicates the relationship between sequences. It does not show the direction of evolution. So we don't know if we have evolved from here to there, or from here to there, or from another starting position on that tree. So we don't know where in this tree did evolution start. Because that starting point, this is what we call a root. It is the last common ancestor in the tree, being the starting point of our evolution in the tree. If we just determine the relationship, we do not know where to start in this tree. We don't know that. There is actually a possibility, there are two possibilities to root a tree, and one, which is the best one, is to include an outgroup, being a very divergent member. For example, if I am investigating a protein families in human, mice, rats, xenopus, and so on, I can include an outgroup, for example, from yeast. Yeast has diverged a long time ago, and therefore we can use this as an outgroup. Of course, we need to find a family member in that species. So it's not always easy to find this good outgroup. But we should actually try to find a family member in a very distantly related organism. If we add that family member, this will actually just place here a root with this being my outgroup. In this way, I know the first split between the outgroup and the rest of my tree is my root. Because this is the most diverse member. So the first split between the outgroup and the rest of the tree determines my root. Of course, if we include an outgroup, we should have a look at the tree if the outgroup is on the correct position. If we find the outgroup, for example, here, then something went wrong. So the outgroup should not be in the middle of my tree. It should be the first split that we have in the tree. A second possibility that we have to root our tree is via midpoint rooting, which is an artificial way to root, meaning that we just look at our unrooted tree and we try to find the middle of this tree, for example here, and then we place the root on that position, and this is the presumed start site of my evolution. This is called midpoint rooting. So midpoint rooting. Which is of course not always correct. Most of the time we will have algorithms that produce a rooted tree, but this is actually produced due to midpoint rooting, not because we actually know the direction of our evolution. If we would like to construct trees, we can actually always choose which type of data to use. So it's possible to construct trees using protein sequences, nucleotide sequences, only coding sequence, and so on. If the goal is to actually determine the relationship as good as possible between sequences, we use DNA sequences. Because here we have more variation available to use to determine the relationship. If we use protein sequences, a lot of the variation that is available and that actually shows the relationship between our sequences will be filtered out. Therefore, to detect homology, we use proteins or 3D structures, but to detect the relationship, we use DNA because we have more information available to determine the relationship. Does this mean that you cannot construct a tree using protein sequences? No, of course not. So you can always use protein sequences and so on, but if the goal is to have a very clear view of how the relationship is between these sequences, it's better to use DNA sequences. Here there are also other reasons, because of course DNA sequences are not restricted to coding sequence. So also non-coding sequence and so on can be used to determine the relationship between organisms. And this of course we cannot do with protein sequences, because it's always coding. If you look at the different steps that are necessary, you can see that multiple sequence alignment is one of the most important steps that we have before we can actually construct our tree. So we have the multiple sequence alignment, here we have the tree construction. Before we can actually construct our tree, we need to specify a model of evolution. Meaning, we need to think about which method do we want to use to construct that tree. Do we use a distance method? Then we will need a distance-based model to model our evolution. Will we use probabilities? Then we don't need distances. So in this way, this step will actually depend on the tree construction algorithm that you use. Remember, and this is something that will come later, in the end it's important to also evaluate your tree. To have a look at how good, how robust is our tree. If you look at the different models that are available, and here I am of course talking at this moment about DNA sequences, there are a lot of different models available. The simplest model that we have is the Hamming distance. Hamming distance being just the distance, so the number of residue changes that we have between sequences. Then we have different models. So here we're talking about DNA sequences. So we can just depict the number of sequences, or we can create a correction, being that it is possible that we have multiple mutations on the same position. Then we only see one mutation, but this can be two. So the more mutations we have, the more that we need to correct for these mutations. This you can do, for example, with a Poisson correction, or we can do this with the Duke's counter model. So the Duke's counter model also takes into account that there is a relationship between the number of observed mutations and the actual number of mutations. So this is not the same. We observe less mutations than there are actually happening in our sequences, or have been happening in our sequences. And therefore we need these corrections. And then we have other models, and the other models take into account, for example, differences between transitions and transversions, the GC content, and so on and so on. A special model that we have, and I don't explain the other models because of time restrictions, but there is one model that is a little bit more difficult, which is the gamma shape or the gamma distribution model. The gamma distribution model is a model, a very specific model, which actually says that the evolution or the speed of evolution is not constant over my protein or my nucleotide sequence. Meaning that if I have, for example, a DNA sequence, it is possible that in my DNA sequence I will have regions, for example here, which are evolving faster than other regions, for example this region, which is more conserved. In this model, we say that there is variable substitution speeds over variable sites. It's a little bit difficult. Saying that, for example, here we can have a high speed of evolution and here we can have a low speed of evolution. If we have this variability, we can take this into account into our model of evolution. And this we can do with the gamma distribution. So the gamma distribution allows us to take into account variable speeds of evolution in different parts of our sequences. If we look at this distribution model, we can actually plot the substitution speed on the x-axis against the number of positions having this speed. If every position in my sequence has the same speed of evolution, we will always have a low speed of evolution, but a very high frequency. And this is indicated with this curve, this blue curve. This blue curve indicates that we have a constant speed of evolution on every position. So every position in my sequence has the same speed of evolution, which means that I will have an overall low speed, but with a very high frequency. The frequency being the number of sites having that speed. If there is some variability, this curve will start to shift, meaning that we will have higher speeds of evolution, but with a lower frequency. And then we have this red curve. And if there is a high amount of differences in speed over sites, we will see that there are higher speeds, but with a very low frequency. And this way, this curve is shifting from left to right. As you can see, this curve follows a distribution. This distribution we call a gamma distribution. And if this curve is on the left, this will have a low alpha value. It's strange that they used alpha for the gamma shape parameter, but not my problem. So they used an alpha value, and this seems to be a very low one. The higher the alpha value, the more variability we have in evolution speeds over different sites. If we want to determine the relationship between sequences, we can take into account the gamma distribution, and we can try to model this gamma distribution and then use this value in the creation of our phylogenetic tree. We can do this in mega, meaning that you have the multiple sequence. It's not multiple sequence, it's a multifacet of our 5 divergent globin sequences, and we'll try to determine the gamma shape parameter. So I'll import my 5 divergent sequences. So I close my data. I'll do align, aided build alignment, retrieve sequences from a file, and here I have my divergent globins. I select open, and I'll align my 5 globin sequences using muscle. So don't forget to align them. So I'll align them using muscle, and then I can send them for phylogenetic analysis. To determine the gamma parameter, the gamma shape parameter called alpha, we can use the ratio button, and there we have estimate the gamma parameter for site rates. As you can see, also here we make use of a model which is called the GTT model, which is a good default model. It's an adjusted version of the Van and Blossom matrices. It's an updated version that we have here. You can see that we will try to estimate the gamma distribution, and we will have 5 discrete possible gamma distributions that we will check. So we leave this default on 5. Next we select compute. Let's hope this will not take too long. You will see with the 3 construction algorithms that there are some faster algorithms and there are some slower algorithms. Voila. And then you will get as an output the estimated value of the shape parameter of this gamma distribution. In this way you will see that here we have a gamma shape parameter, alpha, being 6.23. What does that mean? Yeah. Very variable substitution speeds over different sites. So some positions in my globin sequences will have a lot of substitutions, other positions will have a very low frequency or a very low speed of substitutions. So we have high evolutionary rate differences over sites. So a value of 6 is very high, meaning that we don't have uniform rates of evolution. We can use this value for our further analysis. So if we construct a phylogenetic tree, it's always a good idea to first determine the gamma parameter and then use this value in our tree construction algorithm. If you look at the methods that we have to construct a tree, there are 4 important methods. We have distance-based methods, which of course use distances, for example, from pairwise distance matrices. Matrices. Then we have maximum parsimony methods, which try to give us the smallest trees. Maximum likelihood methods will give us the trees with the highest probability. And then we have Bayesian inference methods, which are new methods, also probability-based methods, which are used more and more, but which are not available in Mega, and that's why we will not investigate them. We will restrict our choice to the methods that are available in Mega. If you look at the distance-based methods, this uses distant matrices to create a phylogenetic tree. Here we have, for example, 5 sequences. For these 5 sequences, we will calculate pairwise sequence alignment scores, and then we will use these scores to calculate distances and use the distances to calculate or to construct a tree. Here, with these distance methods, we try to find a tree in which the branch lengths correspond with the pairwise distances that we have between sequences. So if we perform pairwise sequence alignments and we can see a distance between sequence A and B, we want to represent these distances in our branch lengths in the tree. This looks very similar to the progressive methods for multiple sequence alignments, because also here we start with placing the 2 most closely related sequences together, and then we enter a 3rd one and a 4th one and so on. So we will always take the 2 most closely related sequences together and together and together and together, and in this way constructing our phylogenetic tree. But of course, here we use distances. These are very fast methods, meaning that we can use them on thousands of sequences at once. So to construct a tree with a very large number of sequences, we can use these distance methods. But of course, they are not always the most correct methods. because they are distant methods. There are two important groups, UPGMA and neighbor joining. You should know these two groups, so it's important to know the difference between these two. And if you look at this, we can see that, for example, for UPGMA, unweighted pair group method with arithmetic mean, you can see that all the OTUs, so all our input sequences, are on the same position. Meaning that here we actually think that every sequence has had the same amount of evolution to the root. So the distance from this one to the root and the distance from this one to the root is the same distance. So every sequence has had the same amount of evolution. Of course, and you can already notice that this will not be correct. So in real life, not every sequence has the same amount of evolution as another sequence. Some sequences will evolve faster, will accumulate more mutations, others will evolve slower. But here, it actually states that every sequence has the same amount of evolution. Here we automatically, for UPGMA, create a rooted tree. So this is something special. UPGMA automatically creates a rooted tree, because here we determine the direction of evolution. On the other hand, another distance-based method is the neighbor joining method. And the neighbor joining method is different, and you can already see this. They are not all on the same position, meaning that the amount of evolution is different for every sequence in our tree. So they all evolve with different speeds. They have all different amounts of evolution. Also here, and this is wrong in this tree, this produces an unrooted tree. So the default tree created by neighbor joining is an unrooted tree. So it's actually better to represent these results in a star-like tree instead of this rooted tree. So here we see a rooted tree as a result, but the default tree that is created by neighbor joining is not a rooted tree. So this should better be a star-like tree, or they used midpoint routing, or they use, for example, an outgroup. This is also possible, to root a tree. So it's not because neighbor joining produces an unrooted tree that we cannot root it. So to include an outgroup or to use midpoint routing, we can still root the tree. Next method that we have is maximum parsimony. I already indicated that maximum parsimony will provide us with the smallest tree, being the tree with a minimum number of substitutions that are necessary to create a tree. And this way we will always get a rooted tree. So maximum parsimony produces a rooted tree. Always. Because we get the tree with the smallest amount of substitutions that are necessary to create a tree. What does parsimony mean? For example, I don't know if you're from West Flanders. No? If you go, let's do it a little bit different. If you go from Bruges to Ghent, there are multiple ways to go from Bruges to Ghent. You can just take the E40 from Bruges to Ghent, or you can take all the small roads that go via Maldegem, Eeklo, and so on, and go to Bruges. This is also a possibility. So there are multiple hypotheses for going from one point to another. This is also the same for evolution. There are multiple ways to actually look at evolution. It is possible that one residue is mutating into another, or this can be a multi-step process. That I first mutate my residue into another one, and this is mutated into another one, and mutated into another one, and this is very complex. Maximum parsimony actually says that we should investigate all possible hypotheses, but that the simplest hypothesis that we have is correct. So if I ask you, and you came from Ghent and you go to Bruges, how did you get here? I know, using maximum parsimony, that you just take the E40 and go in a straight line from Ghent to Bruges. This is maximum parsimony. And this is also an evolution. We create all possible trees. In these trees we look at the number of substitutions that are necessary, and the smallest tree will be the most parsimonious tree and be the correct tree, because we need a minimum amount of evolution here. This means, because we know how many substitutions that we need, that we also know the direction of evolution. And that's why it's rooted. A problem that we have here, because we have a minimum evolution tree, is that the tree that we have looks a little bit the same as our UPGMA tree. Meaning that it will place all OTUs on the same position. Every OTU will have the same amount of evolution. And then we have a problem. This is called long branch chain attraction, that if we have one sequence that has evolved very fastly, that it will not place that sequence on the correct position. Because it will try to, how do we say that? Divide the substitutions that are in that branch over the whole tree. And that's why it will place that sequence inside my tree on a wrong position. This is called a long branch chain attraction artifact. Because it wants to place all sequences on the same position in my tree with the same amount of evolution. It will also place these sequences inside my tree on a wrong position. So this is what maximum parsimony does. So this is a drawback of this maximum parsimony method. The last algorithm that we have is the maximum likelihood algorithm. The maximum likelihood algorithm is a non-distance based method which actually calculates the probability of observing a tree. This can be done, of course, with a model of evolution. So based on a model of evolution, it can calculate the best tree based on the observed data. In this way, every residue has a likelihood of being on that position in my multiple sequence alignment, and these probabilities are taken all together in a model to actually determine the most likely tree. So this is some probability calculations that we have here. Of course, we have a lot of different algorithms available, and it's not always very clear how to determine which algorithm to use. And therefore we have this type of workflow. So we start with creating our set of homologous sequences from our protein of our gene family. Then we create our multiple sequence alignment, and once we have our multiple sequence alignment, we will have a look at the similarity. We will have a look at the similarity and we will see how strong is this similarity. If we have a very strong similarity, meaning I can give you these input sequences and you can create the multiple sequence alignment yourself without the help of a model of evolution, without the help of a computer, this is strong similarity, we can actually use maximum parsimony as the best method to create a tree. Because here we know that every change that we see will be the result of one substitution and not two. So here the simplest hypothesis will be the correct one. If we have recognizable similarity, meaning that if you can create this alignment yourself, but there are some regions where you need some help to actually align them, there it's better to use a distance method. Because in this distance method we already have information from evolution that we can use to aid the construction of our multiple sequence alignment. If you don't have any recognizable similarity, then we are a little bit desperate, and then we only have one possibility left. We just use probability calculations. So there we use maximum likelihood as the method of choice. If you look at, for example, the divergent globin sequences, how would you describe the similarity? So if you look at the multiple sequence alignment of these divergent globin sequences, how would you describe the similarity? Is this strong, recognizable, or not recognizable anymore? Not recognizable? Not recognizable. Because there, we can not even start to align these sequences. You can try, but you will not find a position where to start the alignment and you will never get a good result. Therefore, for these sequences, it's better to use a maximum likelihood tree construction method to create these sequences. After we constructed our tree using neighbor joining, UPGMA, maximum parsimony, maximum likelihood, or whatever, we need to evaluate the tree. There are multiple possibilities. You can have a look at the accuracy, the consistency, the efficiency, the robustness, and so on of the tree construction algorithm, but we will not investigate that. There's only one thing that we are interested in, and this is the robustness. How robust is the topology, so the relationship that I see in my tree? So this is about the robustness of the tree topology. What if there would be only half of the information? For example, I have a multiple sequence alignment of 100 residues. What if I would only have 50 residues? Would I still have the same tree? Or what if I would have more information available or less information available? Would I still have the same topology, so the same branching order in my tree? So this is what I want to try to evaluate. Not if this tree is correct. We cannot evaluate that. So we can never evaluate if this is correct. It depends on the algorithms that you choose, it depends on the input that you have, and so on. The parameters that you set, so we can never detect that. But we can detect the robustness of the tree. So can we actually create that tree over and over and over again, even with more or less information that is available? This we can do with non-parametric bootstrapping. What we actually do is not create that tree once, but create that tree multiple times. And normally this is done between 100 and 1000 of bootstrap replicates. So we will try to create our tree, for example, 1000 times. As a default value, it's good to have 500 bootstrap replicates, because it's somewhere in between 100 and 1000. There will be some methods which are a little bit slower, and then we will lower the number. For example, a maximum likelihood method is slower, and there it's maybe better to use 200 bootstrap replicates, but you cannot go below 100. So use 500 as a default. If you see that it is very slow, you can stop it, and you use a lower bootstrap value, for example, bootstrap 200. How does this bootstrapping work? It will actually create, because if we have a multiple sequence alignment, for example, this is my multiple sequence alignment of 4 different residues. If I use a tree construction algorithm for this multiple sequence alignment, and I do this 500 times, I will have 500 identical trees. Which means that my robustness is perfect, because I always have the same tree. Therefore, we need to create artificial datasets. And for these artificial datasets, we actually create a new multiple sequence alignment by randomly choosing columns from my original one. In this way, I don't invent new information. So it's the same information that is available here, but I choose the information randomly. It has to be the same length. So it has to be the same size. Meaning, if I have here 4 residues, I need to create an artificial data set with 4 residues. I cannot make it shorter, I cannot make it longer. But I can randomly choose columns, meaning that I can choose, for example, as the first one, this as a second one, then the first one again, and then maybe the last one. And this way I can lose some information, and I can take some information into account multiple times. So this will be more important, and this will be, or will not be used anymore. And this way it is actually possible to create new trees, different trees, with the same multiple sequence alignment, but focusing on different positions, more or less, in my original multiple sequence alignment. So this is possible. What is the result? If I look at my resulting tree, I will get some frequency values in percent. Meaning that for my original tree that I created, my first tree, this will look like this, which is my best one, but then it will actually indicate how many times that I have these 2 sequences together. So here we distinguish a clade, being these 3 sequences, and on the position of that node it will indicate how many times can I find these 3 sequences together in one clade. Here this is 95% of the times. Here we can find these sequences, these three sequences together 90% of the times. We need 70% or more to support or clade assignment. Meaning if this value drops below 70%, we are not sure that this is the correct branching order for these sequences. So this can be a problem. Let's try this. We already performed the exercise for the different ALDP proteins. We saved it as abcd.fasta. And now we will try to create a phylogenetic tree for this family. Before we create a phylogenetic tree, we need to do two things. What is the first? The gamma. So we will determine the gamma shape parameter. And what else? Before we can determine the gamma shape parameter? The multiple sequence alignment. So first we will perform multiple sequence alignment. Next we will determine the gamma shape parameter, and then we can try to use this gamma shape parameter in our model. Let's have a look. So I'll open my ALDP proteins, so my ABCD proteins. So edit build alignment, retrieve sequences from a file, and then I can select my ABCD as input. Before I can align these sequences, maybe I can shorten these names. So this is my ABCD 1 from Homo sapiens. This is my ABCD2 from Homo sapiens, and so on. And the last ones I will call PXA1 because they are from yeast and they have this name. So in this way I have given them shorter names. If you would like that, you can actually assign a group name to sequences. For example, this is human, also this, this, this is mouse, you are not obliged to do this, so you don't always need to provide a group name, but it is something nice to actually show. So yeast, voila. Now I will create my multiple sequence alignment, so I'll use my muscle align protein and compute. And now you can already try to answer the question about the relatedness of these sequences. How similar are these sequences? Is this high similarity, recognizable similarity, or no similarity? Would you indicate this as high similarity? No. With high similarity, you should think that I can give you these sequences on paper and you can create this yourself. I don't think that you can create this yourself. So here we have there is similarity, so we recognize that there are some columns that we can actually create ourselves, but we cannot create everything ourselves. So it's not high similarity, there is some similarity, but not high. Is there no similarity? No. So there is some similarity, and therefore we should actually use a distance-based method. So neighbor joining or UPGMA. The difference is if we use neighbor joining, we have branch lengths, and with UPGMA we don't have branch lengths. The problem with neighbor joining is that it is unrooted. With UPGMA it's rooted. So it's up to you. So I made my multiple sequence alignment. Now I can send this for phylogenetic analysis. And in the next step I can determine my gamma shape parameter. So I use to estimate my gamma parameter for site rates. I use the same settings as indicated here. And I'll select compute. If this takes too long, I will stop it and will just use uniform rates. Nee. Dat is niet uit.
Oké.
Nee. Dus de vergelijkende formules die er staan, zo voor Hamming distance en Poisson correctie, is niet. We weten wat is, hè? Dus dat wel. Maar je moet niet de formules hebben. Nee. De formule van Hamming distance is wel zeer simpel. Het is een aantal verschillen. So here we have a gamma distribution with a gamma parameter alpha. The alpha value equals 3.0949. So we can remember this. Most of the times we'll just write it down. And now we can use this parameter to indicate that we have some variability over different sites in rate. Evolutionary rate. You can keep the results, they are here. So if we select this opens again. And next we can start to construct phylogenetic trees. So the best way is to use a distance-based tree. For example, let's start with UPGMA. With UPGMA, this is a distance-based method which is also fast. So here we can indicate that we want, for example, 500 bootstrap replicates. So it's normal that these settings are different. So we need to indicate that we want to use a test of phylogeny. As a test of phylogeny, we use bootstrap, and we always use a test of phylogeny. So this is very important. You always need to use a bootstrap method to evaluate the robustness of your tree, always. For a distance-based method, we can use 500 replicates. We can use the GTT model, and here, rates among sites, we don't use uniform rates, but we change this to gamma distribution, because we just determined the alpha value. As a value here, we can indicate that this is 3.0949. So we can actually enter the gamma parameter here, the alpha value. So 3.0949. The other settings we keep as they are, and then we can select compute. And you can see here that this goes very fast, so these are the number of bootstrap replicas that we need. While this is running, you can already start a second. Well, of course, it takes memory on your computer, so this will slow your computer, but you can already start the next tree construction method. Maybe, for the time that we have, we can reduce the number of bootstrap replicas. So I'll start a new one, for example, neighbor joining. Here we can reduce this, normally you keep it as 500, but we can reduce this to 200. GTT, gamma distribution, 3.9049, compute. We can also try the other ones, so phylogeny, we can use maximum likelihood. For maximum likelihood, I'll put this at 100. It's the minimum, so normally you take a little bit more. GTT. Here, of course, gamma distribution. But, and this is important, if you look at the setting for maximum likelihood, here we do not enter the gamma parameter. This is different. Here it says the number of discrete gamma distributions, or gamma categories. So this is what we actually filled in to determine the gamma parameter. So here it will calculate the gamma parameter again. So here we just keep this at 5. So don't fill in the value of the gamma parameter because it will calculate it himself. This is for maximum likelihood. Then we say compute. And now maybe we can start the last one. The last one is maximum parsimony. For maximum parsimony, again I'll use for today bootstrap 100. Substitution type amino acid, gaps missing data, complete deletion, and you can see that we cannot provide a gamma distribution here. So the maximum parsimony method just takes into account the number of substitutions and does not take into account variable size. And we can select compute. This goes faster than expected. So for maximum parsimony you can use a higher bootstrap value. Let's have a look at the results. There's always a good caption, so this is very nice to use. You can see that there's also a scale bar if you scroll. And what can we see? Which method is this? Neighbor joining. So this is a neighbor joining method, giving us, and you can already depict this because the size of the branches is different, they're not all on the same position, giving us an unrooted tree. Here the tree seems to be rooted. How is this possible? What happened here?
ABC4 is used as outcome.
Yes. No. It's not correct. It's not because they use ABCD4 as an output. It's something else. How did they create the root here? Midpoint routing. So this was done with midpoint routing, and in this way they actually depict this clade as some kind of outgroup. Because we don't have an outgroup.
Ah, okay.
We did not include one. No, it's just because it's still rooted, it uses midpoint routing. As you can see, for the clade assignment, we know that from our, you can see 200 replicates, these two sequences are together 100% of the time. These are together 100% of the time. 100, 100, 100. These are together 44% of the time. How is this possible? So only in 44% of the cases I can find these sequences together, but if I look at these values, they are together 100% of the time. So what happens here? How is this possible? Is it possible, for example, that this group is somewhere between these groups? Is this possible? So here we have a group, group, group. Is it possible that this will be mixed within these two groups? No. Why? Because they are always together. So that is not possible. You cannot place this inside there. What can happen? Yes. This can be somewhere else in the tree. So it can be combined with, for example, these yeast genes. It can be combined with the ABCD4 genes. But it will not be combined with this group, because this is always together. And in this way we can always have a look at how this is combined. If they are always together, and they are together in 36% of the time, then we also know this one. If this is 100, then we know that they are always 100% of the times together. So here we have a bootstrap value, which is not shown. This should be 100. Because we know that this is 100. Then we also know that the rest of the tree should be 100% of the time. How this is organized in that 100% we don't know. But this is how you can see this tree. A better representation for a neighbor joining tree is not this rooted tree, because of course here the root is something guessed, it's fictive, is to actually depict this with another visualization. For example, to use a radiation. So you can depict this here, you can select there, and can select radiation. And in this way we can actually change the format of our groups. Of course this is a little bit too big, so now I can use this zoom icon. So I click it, and then I can drag it on my screen. And this is actually a better representation for my neighbor joining tree. Of course, because I need to make it smaller, it is more difficult to read. But this is a better way to visualize this neighbor joining tree as an unrooted tree. Let's have a look at the second one. The second one is my maximum parsimony tree, UPGMA tree. And here in the UPGMA tree I have the same divisions as last time, but have a look at the bootstrap values. They are a lot better. So here I have enough information to actually say that we have this organization of the tree. Let's say it in that way. It's the same organization as we saw with the neighbor joining method. Is this a rooted or an unrooted tree? It's UPGMA, rooted. So here we actually have a root because this method actually says that we start here and in this way we split up. So here this is automatically a rooted tree. And we can also see that they are nicely at the same position. So of course the branch lengths we cannot use for any other purpose. So here the branch lengths are not that informative, because every branch has the same amount of evolution here. But the bootstrap values are better. So this would be, in comparison with, for example, the neighbor joining tree, a better tree because we have better bootstrap values. Here we have the maximum parsimony, and I think that let's have a look. I cannot see it. I think that my maximum likelihood tree is still running in the background. This is my maximum parsimony tree, indicating that this is the tree with the minimal number of substitutions that are necessary to produce this tree. If I look here, I can see A, B, C, D, 1 and 2, but then suddenly I can see that I have my PXA2 here and there. If I look at the assignments, this is of course something different, because here I have values below 70. So this will not be that good of a tree. This is something strange. Watch out with maximum parsimony, because it is possible that you have multiple trees that are even, or that they have the same amount of parsimony. Meaning that it is possible that we have multiple numbers here and multiple trees as a result. Not just one tree, but multiple even parsimony trees. So this is not that good. Of course, the number of evolution that we have between our sequences is too big. Should this be rooted or unrooted? This is rooted. So this is a rooted tree because here we know the direction of evolution, because we needed to count the number of substitutions that are necessary to create this tree. And I don't have the results for the maximum likelihood. It's still busy. Which is, it's doing something. For the last part, I will not discuss this all. This is a more theoretical part, discussing genetic variation, discussing SNPs most of the times. The most important part that we have here is the exercises. So if you want to focus on this, focus on the exercises that we have. Most of the times this will be about the DBSNAP database and about the OMIM database, which are linked together. So this is the most important part for this last part. There's also an indication of the next generation sequencing pipeline. So this is as some kind of introduction to next generation sequencing analysis, which we will see in high throughput analysis. This is just an introduction, so don't focus too hard on this next generation sequencing pipeline. I am more interested in the link of SNPs that are available in genetic diseases. So if you know this, I think this is the most important. Just exercises, just only focus on exercises here. Don't focus on the theoretical aspects behind the information that is available for this last part. So this is just a hint for the exam. Focus on the exercises, don't focus on the theory that is available here. Do we have the maximum likelihood tree? No, I'm only at replica 15 of 100. So this will be for a later day. What is important for maximum likelihood? Will this produce a rooted or an unrooted tree? Unrooted. So this looks a little bit similar as neighbor joining. So this will produce an unrooted tree, and therefore also the visualization is better in this star-like tree than in a rooted tree version. Just to give you this information for home. For the exam, I can only emphasize the exam or the organization of the exam is depicted on the homepage on LEGO, which is very important to know. It's depicted below. I also showed this the first lecture, which means that we have 3 parts. A first part will be a knowledge and insight exam on your lockdown and lockdown browser software. You use this in another exam, lockdown browser. Okay, so this is good. Still keep lockdown browser, you will need it. Second part is more insight, so this will be exercises, the second part. There you will have access to all applications, but it's not open book. So it's closed book, but you have access to the applications that you need. You have access to the internet and so on. This is about 40% of the score. Part 3 are questions related to the assignment, which I also gave in the beginning. This takes into account for 20% of the exam. Know that you should be able to make exercises with the software that you installed on Linux, but you should also be able to explain, for example, how you installed the software. So both parts can be used, and this is open book. So everything that you gather of information, every note that you make, you can use to answer these questions. Okay? Then I wish you good luck.